Thanks.
Time as any, Mr. Newman. Take her down to periscope depth.
Yes, sir. Clear the bridge. Dive.
Stand right.
Greetings. Welcome to the Evidence and Reasons video channel. I'm Salvador Cordova, and today I'm privileged to be joined by Peter, and he sometimes goes by the handle CryoDebunk, which I think is a really cool internet handle. He was very kind to join me in two previous shows where I tried to respond to his questions. He was a co-host, and I felt the questions he raised were very important, things that creationists should study and learn. And I'm privileged to have the chance to try to explain some of the reasons I believe things. One reason that we have the Evidence and Reasons YouTube and video channel is that the Christian faith, if we look at the New and Old Testament, it teaches that God is a hidden God. And one of the reasons that he is hidden is that he will shame those who are wise and intelligent in this world who reject him, and so that God will get the glory. Now that doesn't seem like a very nice thing to do to people, and I admit that. And yet on the other hand is, unless there's any evidence to even think that in the first place, why believe it? So I think that the Bible at least does give an answer why a lot of evidence and reasons are hidden. On the other hand, as I have studied the topic of origins, the origins of the universe, the origins of life, the origin of eukaryotic organisms, etc., it's given me reasons to believe that there is a creator and an intelligent designer. And it also teaches in the Bible that that it's the glory of God to conceal a matter, it's the glory of kings to search out a matter. And I interpret that to mean that with some degree of study in God's providence, that we can see and find evidence for God that may be hidden away, but with some effort can be discovered with providence from God allowing it to be discovered. So that's a long opening for why this channel is organized, and why also I spend quite a bit of time combing through biological literature, physics and chemistry, and theoretical population genetics, theoretical evolutionary genetics. So I believe that in the process of doing this, we'll see that life is a miracle. But wherever anyone is in their beliefs, I'm just going to kind of step aside from that for now and just try to answer things by appealing to what the present literature says, and then some considerations. And so let me share my screen. There were some questions that Peter
Yes.
had raised, and I'm going to put it up here. We were going through a list of 6 questions, and for the interested viewers, part 1 and part 2 covered the first 2 questions. Today we'll try to cover questions 3 through 6. I may not cover it in order. So question 3, generally, how do you figure out if some mutation is slightly deleterious? Question 4, what is your definition of Turing completeness? And do you think that a system must be Turing complete in order to be able to reproduce? Or I understand it not correctly. Question 5, what do you think? The peacock tail is negative to fitness? And number 6, what are your best examples of irreducibly complex features? I think that number 6 is kind of an easier one to answer at this time. And I'm going to try to go through all 4 of these questions today. And I really want to thank Peter for his patience and giving me a chance to spend practically 3 or 4 hours answering his first 2 questions. And that was a lot of fun for me personally. So what are my best examples of irreducibly complex features? I would say the translation and transcription system of DNA, and I'll show some parts of it. So let me see, I'm going to bring up, see if I can bring up a video. So for origin of life, which is the phase before the what we call the biological evolution phase, I think the origin of the transcription and translation system we might call irreducibly complex. Just for the record, I don't like using that term at all. I would just say something that is not explained well by natural selection, random mutation and natural selection. One reason that this is particularly problematic for origin of life scenarios is that before there is a genome or a replicator that uses a genome, natural selection can't be in operation. So let me see if I have I'm going to show a homohexameric helicase. Let me bring that up and then I'll see.
Separate nucleic.
Okay, so let me try to, this is going to be a little loud, so I'll turn it down just a bit. And let's see here.
Acid duplex.
And so now I'm going to share my screen and audio. Share screen, audio.
Helicases separate nucleic.
Peter, could you hear that audio in the
Yes,
video?
I hear it.
Okay, great.
Yes.
We'll go ahead and watch it.
Helicases separate nucleic acid duplexes into their component strands using energy from ATP hydrolysis. The crystal structure of this DNA helicase from bacteriophage T7 reveals a hexagonal arrangement of 6 identical subunits. Surprisingly, the ring is not 6-fold symmetric, but is slightly squished. A model for the mechanism of how the enzyme might work explains this structural asymmetry. Of the 6 potential ATP binding sites, 2 opposing ones bind ATP tightly, 2 are more likely to bind ADP and phosphate, and 2 are empty. These 3 states may interconvert in a coordinate fashion as ATP is hydrolyzed, creating a ripple effect that continuously runs around the ring. Because of these conformational changes, the loops that extend into the center hole of the ring that are proposed to bind DNA oscillate up and down as seen in this cross-section. The oscillating loops might pull a DNA strand through the central hole, thus unwinding the double helix in the process. A frontal view shows the full dynamics of this fascinating protein machine.
So turn micro.
I'm sorry.
Okay.
Go on, Peter. I'm sorry.
Listen, I just you wanted to say something.
Yes, I'm sorry. I was muted. I didn't realize it. So I'm going to bring up a slide here, and I'm going to show something about the helicases. So it's not just the helicase. It's the DNA and the transcription and translation system. So helicase and DNA are only part of the entire system, and I'm just showing 2 critical parts. The DNA is needed. I'll try to explain why. See if I have it here. I actually have the spelling of the helicase somewhere. Let me see if I could find it. Yep. Okay. I have it here. So I'm going to explain this for the viewers here, and I'll turn off the notes. This here is the listing of the amino acids in helicase. It is each of the letters represent an amino acid. So this is I just took this from a protein data bank. Now, for the helicase to work, as in the video, it actually has 6 copies each of that. It has to generate 6 copies, 6 exact copies, and they have to be located and assembled, and then it can start operating on the DNA. So if we have these 6 copies here, this would violate random chance assembly, because that would violate the law of large numbers, or however way anyone wants to phrase it. It's just highly improbable that this would happen by chance. One way that the chance is circumvented is if there's an existing genome, like such as DNA implementing a genome, so that the recipe for the parts of the helicase can be repeated through a process of transcription and translation. On the other hand, so a helicase needs a genome as a matter of principle to exist in order for the helicase to function. Now, people will say, well, origin of life researchers, no one believes that something as complex as a helicase, the current day helicase was needed to unwind DNA, to which I'll respond, I said, well, that's true, they may believe it, but they're appealing now to things that don't have any direct observation or experimental evidence. They're appealing to something that they can't prove, and they're not even able to theorize what the alternatives are. I mean, there are helicases that don't have 6 units, maybe as small as 4, but there's still a problem of requisite symmetry. I think I tried to point out that even in the man-made world, symmetry is very useful for doing things. There's a reason that, there's a physical reason it makes sense that helicase would have symmetry. Now, as I said, there are other kinds of helicases that have only 4, but it's still a symmetric system. So the problem is, to answer Peter's question, what do I think is the best example? I think it's the transcription and translation system. So helicase needs some sort of genome to implement it. And if one appeals to a genome other than one made by DNA, then that creates another problem, and then an appeal to something that is not directly scientifically testable. And we wouldn't even know how to construct it. I've talked to biochemists, and some of them said, no, we wouldn't even know where to start unless we had a working model like this. One would think that something of only maybe 1,000 letters, I mean, that's like, what, 5 comments in a chat of a YouTube stream, you think someone would be able to type it out, but it actually turns out to be a pretty difficult problem in solving protein folds. So, you know, if we have some software that if we throw a whole bunch of letters at it, it will estimate the protein fold. What we're not able to do is to say, okay, let's just start from scratch and build a machine, now generate the letters to create the appropriate folds, because these things have to have very, very, very tight tolerances. And there are other machines out there that are even tighter tolerances in terms of the size. It's just like the problem of making a bolt that is the right size to fit a screw or a nut. So then the other problem is DNA can't replicate without something like a helicase. The helicase unwinds it and enables replication. So let me just show that again a little bit, just for the reader's benefit. I'll turn down the volume. So here's the DNA here, and for it to replicate, the helicases are here. It's going to open it up, and then replication can happen. So that, for origin of life, that is my favorite example of quote-unquote irreducible complexity, although I don't like using that term at all. I think it's been one that's been abused. For eukaryotic evolution, let me see if I have it. I have it on another slide. Hang on. For eukaryote, and I'm actually hoping I'll have a chance. I got word that I might want to be paired up against Rna. And this is what I'm going to use in my debate with Rna, if he accepts the challenge. Let's see if I can get this here. For eukaryotic evolution, it's this one. Now this is going to take a lot longer to explain. There are not any dynamics, but the basic issue is that without a lot of codes put on the proteins, what we call localization signals, both on the RNAs and the proteins, it is statistically improbable. From a practical standpoint, it's statistically impossible that a prokaryote like a bacteria is going to evolve into a eukaryote like yeast. And this is a very long, involved discussion, and one that I might be having a totally separate, say, 2-hour video on, because it is intricate enough.
Yes. I do not quite understand what is described here.
Oh, yes. So this is going to be a 2-hour show. So you asked my favorite ones, and this is my second favorite ones. So my first favorite one is the transcription and translation system. My second favorite one is the eukaryotic nuclear import and export system, but that's a 2-hour show. So, but I at least wanted to answer it directly. What are my favorite examples? I didn't explain why that last one was my favorite.
Okay, yes, that's fine.
And I do want to have a 2-hour show. I may invite Dr. Dan in that discussion. I try to invite, for something that technical, I'm glad if it's not one-sided. And I'm hoping that, and now that I mention it, there are probably some discussions where, like what we had for like part 1 and part 2, if I have future discussions like that, I'm going to try to invite someone like Dr. Dan just to say, okay, that's, I wouldn't teach. He can say, all right, Sal, this part's okay to teach in biology class. And then if I say something, he said, no, he's not okay with that. That's fine. I want him to be able to say, okay, this part is teachable in biology class. This part he wouldn't feel comfortable with. And so, so now that I think about it, now that he also expressed interest for the first, to my knowledge, the first time was like last week that he wanted to participate in these discussions. I think that'd be a great thing. So then it'll be a little bit more balanced. And I also do want to hear what he has to say since he is a biology professor. So that's question number 6. What are your best examples? Let me see. Try number 4. What is my definition of Turing completeness? And actually, that's such a good question. I'm going to have to look it up because I actually don't know it. I kind of intuitively know what Turing complete is.
I know it.
Oh, you know it.
I know it from the IT background. There is a clear definition what is Turing system, what is Turing complete.
Okay, so I'm just going to refer to, there are Wikipedia articles on it, and perhaps I'm not going to therefore belabor it, but I'm just going to point.
Can you maybe share screen so I will read it and I say.
Oh, I'm sorry. I forgot to share screen. Thank you. What would I do without you?
Yes, that's so I think.
So here it is, Turing completeness, just the Wikipedia article. So I just invite the readers to look at this. And
okay, scroll a little bit down, please, because it says nothing.
Let's look at the formal definition. In computability theory, several closely related terms are used to describe the computational power of a computational system. So Turing completeness, a computational system that can compute every Turing computable function is called Turing complete, or Turing powerful. Alternatively, such a system is one that can simulate a universal Turing machine. So this is,
yes, this is a little bit confusing because they do not define what it is. They
yes.
say just Turing complete is Turing complete and says nothing. But yeah, I can say my definition is, may I?
Yes.
So in order to be Turing complete, there must be some equivalent of algorithm what can make forks. If else forks means if something is, if there is some condition, I will go in one path. If in other, I will go in other path. There must be go to jumps and variable assignments. That's what makes Turing complete. And actually, Turing complete models are just, I don't know how to say. Maybe you scroll down a little bit, maybe they will show exactly what it is, because it is not a real definition.
If I could say, maybe not the most formal definition, anything that can run a computer language, that can implement a computer language, I would say is Turing complete. And I overheard one professor say that if you have a machine that can implement one computer language, you could use that computer language then to write a program that'll implement other computer languages. So if we have like the C programming language, we can build an algorithm that will implement any other computer language. You could start with one computer language and then implement it if you have a Turing machine. It's just kind of a funny property.
Exactly. But there is other languages like context-free languages what are less powerful than Turing complete, and you must clearly distinguish between them because
Yes,
so
yes.
yes, we are talking about the system what can copy itself. I'm not certain now that it must be Turing complete. Maybe just some regular automaton what always copies itself, and it must not fulfill all Turing complete features. But maybe I'm wrong. I wanted to hear from you.
Sure, sure. So do I think a system must be Turing complete in order to reproduce? That was your question. I would say no. It doesn't have to be Turing complete to reproduce.
Okay. Yes.
So is a cell Turing complete? I've looked at the literature and there's no uniform agreement on it. Some people will say parts of the cell are, and so I would just suffice to say that let's just assume it's not.
No, I would say as a complex cell like eukaryotic, multicellular, or so I would say they are for sure Turing complete because there is branches. Either cell do one thing or either it does some other thing that makes Turing complete. And what about the viruses? Virus, it's like it is doing always the same. It copies itself, but it does every time the same thing, so it's not Turing complete.
Yes.
It's my broad definition.
Yeah. I mean, I would lean toward it, but I would lean toward saying the cell is a Turing complete machine, but I would not be, I would feel uncomfortable saying absolutely. However, that being said, one evidence of that it's Turing complete is there are people that are trying to make computers out of cells. So it has at least a lot of the hardware. They call it DNA computation. Let me see, just to make sure I'm not making this up, just even for my DNA computation. Yeah. Okay, so DNA computing, another Wikipedia. DNA computing is an emerging branch of computing which uses DNA biochemistry and molecular biology hardware instead of the traditional silicon-based computer technology. So I've heard of this, and they actually have to do it really well, they have to use pre-existing cells. So the cells already have a lot of the machinery to do this. So if it's not Turing complete, it really has a lot of the machinery already there. So,
can you tell just broad what do they do there? Because it's just generally interesting.
Yes, one reason they're looking at DNA computing is they're trying to make computers that are smaller and smaller. Mm-hmm. And there's some advantage to having a DNA storage, because if you have many cells, there's a way to actually just make so many copies of something, it's automatic archiving. And then also it's just kind of interesting. A lot of academics will pursue things that may not have immediate practical value, but it's just an interesting challenge. They're able to solve certain problems in computer science using DNA that might have been solved also with regular silicone machines. And so the other reason that it may be of interest is if they're able to build DNA computers, the pharmaceutical industry might actually be interested in that, where we have basically intelligent robots that could treat our diseases. So even though it seems very far-fetched, it's still just very interesting in its own right, whether it leads to anything. But the idea of being able to create what they call synthetic biology touches on this. And the pharmaceutical and biotech companies are always wanting to tinker with life for various purposes. And yeah, it is a little disturbing. If they make a mistake, we could end up creating all sorts of...
I wanted to ask, do you think that these researchers are sort of positive, or you are against? Because I saw some critics are like against some new researchers, including genetic engineering and other stuff. So what's your opinion?
I think the risk is low, but let's say that something goes really wrong. I think there's evidence, not maybe the strongest, that the coronavirus may have been an accident of bioengineering that got released, either
Yes.
accidentally. And I mean, there's not 100% proof, but let me put it this way. I know the Defense Department looks a lot into bioterror, and also they probably are developing biological weapons themselves, and that's pretty scary. Of course, they're not going to talk about it much, but the technology that is there if they wanted to make something that is deadly, and that does bother me. So it can be used for good, but it can also, I think the technology is advanced enough that it can also be used for harmful, very harmful purposes.
Yep.
So, I mean, if we can start using genetically modified organisms to cure people, it's probably going to be even easier to make genetically modified organisms to harm people.
Mm-hmm.
And we know that there are some examples of genetically modified organisms that are destroying certain crops. So that's a little bit hard to talk about. And my boss was a famous pioneer of genetic engineering, and I don't bring the topic up about whether it's good or bad. I just like, that's a taboo topic. I try not to talk about it even when I'm around him, because I feel it's kind of sensitive. So there was one where they had, oh, here it is. This researcher, Alderman, they were able to solve the Hamiltonian path in a graph with 7 summits. That's a problem in computer science. And then the NP complete problem, they talked about it. There is one that's more mundane but interesting. They call it the traveling salesman problem. Like,
Yes.
yeah. So that was solved by, that's the only one I know that was solved by probably a DNA.
They should say in what time they saw, like, or brackets and whatever. So this is how you present, how you solve the problem, but still.
Yes. So anyway, back to the question, Turing completeness, and I'm going to back to your question. I think we could make a case. I would just not feel comfortable at this time saying it absolutely is. I would feel more comfortable when we saw more peer-reviewed papers saying, affirming that. I just haven't seen them, but there are hints of it. Now, what I meant when you said, do I think a system must be Turing complete in order to reproduce? I'll give an example why I don't think so. So I'm going to show a salt crystal here. See if I can, molecules.
So you mean crystal growth?
Yeah, see, like this, there are some things that repeat as a pattern, and it'll just happen spontaneously. So we don't need a Turing. There's certain things in nature that will just naturally form repeating patterns. It'll reproduce without any need for a Turing machine. So, so the question is cellular life like one of those that it'll spontaneously assemble and duplicate? And to the best, I mean, all that we know is that it doesn't happen. I mean, the creationists will argue it doesn't happen spontaneously, that there has to be the organization of a cell is created by the organization of a pre-existing cell. And we don't see any exceptions to that. And then I would refer the readers to the book. Let me see it. Let's see here. Let me bring it up in another window. It's going to use up a sorry. Let me bring it up in another window here. It is the book by Change Loratan and Rob Stadler. The book is entitled The Stairway to Life. And that book will go into all the reasons they don't believe that life can spontaneously arise because chemical principles indicate that a lot of the materials of life have a much stronger tendency to disorganize, and that only something as complicated as the organization of a cell is competent enough to maintain this, to perpetuate its own self or perpetuate duplicates of itself. It just doesn't happen spontaneously. So let me see if I can just show the reader. Let's see if I can get it loaded. See if I have it here. I'm going to show it as a title. And so the cover, all right, I'm going to put it up here. So this is from the Kindle version. And let me share my screen. So that's the title of the book, The Stairway to Life, James Loritan and Rob Stadler. And let's see, did they have a back cover? I wish they had a back cover. Well, let's just see if there is a back cover. No, they don't have a back cover. Anyway, there is...
Is there some kind of summary also to show?
Oh, so here are the people listed, and you'll see my name right there listed in the...
Yeah.
So it's just a little bit of self-promotion. But the people here who participated, they're very, very smart. Many of them are... there are people on the back cover that were listed who are biochemists teaching at accredited universities, both secular and religious institutions. So I just wanted to share that with the viewers, going back to the question about Turing completeness and the ability to reproduce. That may not need to be immediately answered, but what I will point out, there is a related question which I think is also interesting, and that's the von Neumann. This one's a more, and I'm going to show just maybe a conceptual picture of the problem, because obviously, again, this is of academic interest, but I'll try to explain why. I'm going to share my screen shortly. If we have, and this was of interest to me too, if we built a space probe, it would be really nice that if we could send out a space probe and then the space probe would land somewhere and then be able to build factories, and the factories could keep building themselves, so that when humans arrive there, they have cities already pre-built for them. I mean, that may seem incredibly ambitious, but if we could achieve something like that, that would make it more feasible to colonize other planets, because then machines would just be creating. So it's an interesting topic in and of itself. So this picture relates to, I would presume that this not only has to be a Turing complete machine, but it has to do some other things in addition. So here is an attempt where a lot of the parts, we have like this three-dimensional printer, and what it's going to do is to pull parts from a part shelf here, and it's going to try to construct an exact copy of itself. And then this thing's going to try to make another copy of itself. And they have tried to make some machines that kind of approximate this. What we have found is, yes, we have to supply a lot of the parts pre-made just to help the process along. To be able to do this with like really raw materials of just like ordinary chemicals, that's going to be challenging. So life, the reason this is related to the question of life is that as far as we can tell, life is able to make, it's implementing a von Neumann self-replicating machine. And in fact, in the Wikipedia entries on this topic, it'll mention life. because life is able to do something we can't right now. There's technology in the cell that's able to do self-replication in a way that we can't. So that's another take on the issue of Turing completeness. I would actually say, how about a von Neumann self-replicating automaton? So let's look at the list so far and see. Let me bring up the list here of the other questions. So that we covered my best examples of irreducibly complex features. I already said I don't like the term personally. I prefer saying what are features that I don't believe can arise by random mutation and natural selection, and that's a little bit more direct. And then this thing about Turing completeness, and do I think it needs to be complete to reproduce? I say certain things can reproduce without being a Turing machine. But the issue is, why does life reproduce, and can that organization happen spontaneously? So, I'm sorry. Briefly, what do you think? The peacock tail is negative to fitness. So, the reason that, let me just show a peacock tail. This particular tail, open link. The peacock's tail is very elaborate, and when Charles Darwin saw this, he said, whether joking or not, he did accurately describe the problem. He said it made him sick when he saw this, because if there's competition for reproduction according to the theory of natural selection, then a species like the peacock that uses a lot of inefficient steps, what we call extravagance or almost wasteful steps, should have been selected against. And so Darwin came up with a theory of sexual selection, and I'm going to contact a biologist by the name of Emery Moyna. He has a lot of data on criticisms of sexual selection, but so even Darwin himself saw that this was a problem of it should be negatively fit, because it has a lot of extra baggage, so to speak, a lot of extra steps in the reproductive process. And I don't know if you've, Peter, have seen some of my videos that had Rube Goldberg machines, where it has a lot of added complexity just for the amusement of people, like a machine that would pass salt, or another machine that would open a cookie, and it was just really, really elaborate.
Yeah, this machine, yes, I saw that.
Yeah, Rube Goldberg machines. Yeah. So this problem, probably the most interesting example of this famous experiment is Spiegel. Yes, Spiegelman's monster. So I point the, oh, I didn't know there's a picture of him. Let's see if we have a picture of Spiegelman here. So there's this Wikipedia. Oh, they don't have a picture of him here. Let's see if I can. So I believe this is of Spiegelman here. Yeah, that's Saul Spiegelman. And he was a researcher. And he created what was known as Spiegelman's monster. And so let me back up a little bit. The Wikipedia article, and this does relate to the peacock's tail. Mm-hmm. Spiegelman's monster is the name given to an RNA chain of only 218 nucleotides that is able to be reproduced by the RNA replication enzyme RNA-dependent RNA polymerase, also called RNA replicase. And this monster is named after its creator, Saul Spiegelman of the University of Illinois at Urbana-Champaign, who first described it in 1965. Spiegelman introduced RNA from a simple bacteriophage Q beta into a solution which contained Q beta's RNA replicase, some free nucleotides, and some salts. In this environment, the RNA started to be replicated. After a while, Spiegelman took some RNA and moved it to another tube with fresh solution. This was repeated. Shorter RNA chains were able to be replicated faster, so the RNA became shorter and shorter as selection favored speed. After 74 generations, the original strand with 4,500 nucleobases ended up as a dwarf genome with only 218 bases. The short RNA sequence replicated very quickly in these unnatural circumstances. So this has been advertised as showing how evolution works and that reproductive success is favored. And I would actually agree that this shows that reproductive success is favored, but it shows that what is favored are simpler, are systems that have smaller replication cycles. So something like a peacock that has all these extra rituals to enable the peacock to reproduce should be selected against. Now, I'll point out that bird species on the whole are going extinct at an alarming rate, at an alarming rate. So we're actually seeing natural selection wiping out species now that have long reproductive cycles, and that's their habitats are being destroyed is one of the reasons. It's really kind of sad, honestly. But whereas bacteria, it's like I've searched the literature to see if there's been any examples of whether they believe bacteria have gone extinct, and people have said, well, it's really kind of hard to tell because you can't really search around the world for every location to make sure there's no bacteria there. But we do know bird species are going extinct. So that was that's about the best answer I have to the question number 5. The peacock tail is negative to fitness. Mm-hmm. So peacocks now, some of them have been put, the peafowl is both the peacock's the male, I think the peahen is the female. So the peafowls in general are, some of the subspecies are on the endangered species list because that tail is not really very efficient for flying. It makes them really easy targets for predators. So it is something that is still poses a problem for how natural selection would evolve it, because it looks like it would be selected against. And because there are subspecies of peafowl that are on the endangered species list, it would seem also empirically justified that its fitness is at risk because of its elaborate construction. So, question number 3, how do you figure out if some mutation is slightly deleterious? Now this, I'm saving the hardest question for last, because this is not easy. So do you have any, before I go on to number 3, is there anything I could say more for questions 4, 5, and 6?
Wait a second. So about this Spiegelman monster, okay. So it was, so in the case of selections, the genome was sort of reduced, yes? But
Yeah,
what is about its functionality? What was it actually does? So you see how it's working, because you mean the reduction is not necessarily the loss. Maybe it's good sometimes to reduce it and make it more efficient, because there are useless parts there. So can you tell short but explains it, what did this thing does at the beginning and what it does at the end? That would be great.
Let me try to repeat the question. Let me know if I accurately described your question. What does this illustrate? What does Spiegelman's monster illustrate?
No, not illustrate it. My question was, what does this RNA chain doing at the beginning and the end? So, what is its functionality? Okay, do you understand it? So, what's it able to do at the beginning and at the end?
Oh, okay.
Because
Okay.
yeah.
That's a very good question. We know that there are, we have this thing called an RNA replicase. Now that's actually a protein, it's not an RNA. So let me just see if RNA replicase. See if they have any pictures of what it actually does. Maybe, yeah, unfortunately this is getting really heavy into molecular biology, but this machine here, this RNA replicase, it will only recognize certain RNAs and duplicate them. It's not going to replicate every possible RNA.
May I ask? Okay, they tell that RNA is unstable. So this particular RNA was sort of more stable in order to work by itself just and replicate.
Right. It was unstable. It's basically, we could liken it to each time it got replicated, it had something analogous. We can call it a mutation. I mean, it's not really a total genome, but it's like, so when it got replicated, it had aspects of it that were not identical. It's like a mutation. So the original strand that he started out was 4,500 nucleotide bases long. So he just threw it, and then he let the RNA replicase replicate it. Now these are imperfect replications, which is really astonishing because in a regular cell, if there are problems in the replication of RNAs, there's a lot of machinery to kind of help correct that, because that's kind of bad. Unless there's machinery that allows it for certain specialized purposes, and I'm not that familiar with it, but just suffice to say what he was, again, we're not talking DNA, this is just RNA with a pre-existing protein. And he was, it's just an interesting experiment where if we keep mutating something and we have machinery to copy it, what he found out was as it got mutated, in a pool inside of his like say beakers, he noticed at the end the things that got copied the most were the ones that were only 218 bases.
Okay, yes. And what environment was there? It's also interesting.
Yeah, this was outside of a cell. This is just a beaker, and the only thing that was in it was this. Let's see if I have it. The RNA. It had this. I'm just going to show it for the reader's benefit. It had this molecule in it, like that. It had this molecule in it, and then it had. I'll show pictures for the reader's benefit. RNA. I'm guessing. I could really get in trouble here. Dr. Dan will get on my case. It's either a single or double-stranded RNA. And so an RNA looks like, see if they have this. Let me see if I have a picture of an RNA. So they had RNAs like that, and they had a molecule like that, and then some free-floating nucleotides, which are pieces of the RNA. And other than that, it wasn't in a cellular environment. It was just a beaker.
Yes.
So, I mean, that's about it.
So what was it? It was just the environment with food, so to say, around it. And there were not like special selection to some trait, like resistance to something or something like that.
Exactly.
It was just selection selection just was about the beneficial in reproduction.
Right.
That's particularly why it evolved there. And maybe if there were another conditions there, like there was some danger or something like that, it can be that it has contains parts to fight against this danger and what makes its genome larger.
Well,
Sort of.
I mean, those are good points. I was just showing a very, very simple experiment. Now they have this thing called reductive evolution. And they know that so when it's applied to microorganisms, so this is a case where there's a natural real organism here. So this is from the Wikipedia article on reductive evolution. So this is even more general than Spiegelman's monster. This is something that evolutionary biologists themselves have discussed. It says, reductive evolution is the process by which microorganisms remove genes from their genome. It can occur when bacteria found in a free living state enter a restrictive state, either as endosymbionts or parasites, or are completely absorbed by another organism, becoming intracellular, symbiogenesis. The bacteria will adapt to survive and thrive in the restrictive state by altering and reducing its genome to get rid of the newly redundant pathways that are provided by the host. In an endosymbiont or symbiogenesis relationship where both guest and host benefit, the host can also undergo reductive evolution to eliminate pathways that are more efficiently provided for by the guest. I'll point this out, like the E. coli. E. coli can have a lot of genes, but then there's some that there's some species, quote unquote, or strains of E. coli that have very, very small genomes. They've lost a whole bunch of genes, but they're still able to survive. And so there are examples where it's very easy for organisms to lose genes. Now, microorganisms are special in that they can lose genes and then reacquire them through something called like horizontal gene transfer. And animals don't have it that easy. It's very hard to do horizontal gene transfer between animals. So it does appear that there are, if we can use kind of a more looser term for reductive evolution, there are creatures that have lost their eyesight apparently. When the population moves into a cave and they have no use for eyes, the eyes end up being a reproductive liability, and they end up, the creatures that don't have functioning eyes are able to do better because the eyes don't get infected. And then also the eyes, it takes a lot of metabolic energy to maintain and develop an eye. So it seems, as best as we can tell, that these creatures also have the equivalent of reductive evolution, where they lose capabilities by because of natural selection. What we have had a hard time establishing is kind of getting an estimate of how many actually increase complexity versus lose complexity. And let me see, first law. So Michael Behe put forward a peer-reviewed article in secular literature, and I'm going to alert the readers to it. Experimental Evolution, Loss of Function, Mutation, the First Rule of Adaptive Evolution. And he surveyed all the literature that we have. That doesn't mean we surveyed every organism. So we just had to sample. And he said, he didn't say it in the paper, but in other venues, he said that by his estimate, it looks like 90 to 99% of the beneficial mutations, what we call beneficial in terms of improving reproductive success, is the loss of capability, loss or disabling of genes. And this was, by the way, this was a brutally tough paper to read. I was just so bored to death because he just kept going on and on, citing experiment after experiment. It was pretty thorough, but it was a very slow read. So I'm just referring the readers, interested readers, and you have to be a very special kind of person if you want to suffer through this paper, because it is not an easy read. It was a painful read because he was so thorough in citing all these experiments. So let me see if, I hope it's publicly available. No, I got it because I have university access. So, but he has parts of it available online. So where was I headed with that? So Peter was asking about specifically Spiegelman's experiment. So whatever the flaws of that experiment are, I tried to cite some other papers and ideas that are written by evolutionary biologists themselves to talk about a related phenomenon of where organisms try to get rid of complexity, and how that relates to the peacock's tail is what bothered Darwin is why would such complexity persist for so many eons? Of course, the creationists will say, well, it was created and the complexity's been slowly decreasing over time. And my area of research is that the complexity of the human brain has been declining for several thousand years. And we'll actually be able to measure this decline in human mental capability over into the next several generations. There is concern. I don't know of any major geneticists studying human health that think that our brains are getting better. There is a lot of concern. We're losing mental capability, and then also it's going to affect mental health. There's been a lot of concern that autism has been on the rise per generation. Yes, it could be affected by environmental factors like all the chemicals now and pollutants in our environment that are both affecting our genome, and then even not just our genome, but just our general health. So this is an active topic in medical research, if we are indeed deteriorating, and what can we do to maybe slow it down?
Yes, but the question is, what is reason to that? Maybe just a sort of negative selection, like, you know, if if there is a type of deers, then not the deer what is the smartest are selected by females, but those one with big horns or something like that.
Yes. Well,
so there is yes.
Well, all I could say is that the medical researchers are concerned. Now, there was really...
Yes, I know. I
yeah.
that,
Yeah. So everyone has... there are lots of theories out there, and the creationists have been quick to seize on it. I personally am very concerned about this, and it's one of the reasons I am a creationist. I just... for the reasons I outlined in my talk on Muller's limit, I don't know how something as complex as a human being can, relative to the number of children that each human female is capable of having, I don't know how we can have any chance of improving. I think deterioration is the natural direction, and that accords with the creationist view that we were created and then we've, after Adam sinned, we slowly deteriorated. Now, what I just said is theology. That is not directly science. So I want to make that distinction. So when it's not clear, I'll try to make it clear when I'm talking kind of my own religious beliefs versus scientific observation.
So may I ask?
Yes, please.
Okay. Do you know that there is some human gene that if you break this gene, you do not get fat? So first, is it true, or it is
I don't know, but we do have examples where if a gene is misregulated, it actually helps someone survive. That's the case of what they call lactose intolerant. So Asians, if they drink milk, their stomach gets very upset, whereas the Europeans, they're able to drink milk and it doesn't upset their stomach. It's because the way the gene is said to work in a functional sense is that it's supposed to shut off our ability to drink milk as we mature into adults, but somehow that got broken, and that's what enabled Europeans to drink milk and therefore survive with a different, with a larger spectrum of food sources. Now that's what evolutionary literature says. So I'm not, that's the only case I know where the evolutionary literature has appealed to a broken gene conferring where a broken gene has enabled people to survive better. So just take it for what it's worth. I'm just going to refer it, the readers, to kind of popular. You can Google it, this whole thing with lactose intolerance. And so you asked about if, which gene were you referring to, Peter, that you heard of?
Some gene would make the fat accumulate in body, and if this gene is broken, then that you can like eat calories food, what some people today do, or some other issues with weight, and they do not get thick. And that is, I think it is an example of breaking a gene and make the organism beneficial, because you do not need to accumulate fat. It was need for like if there is like less food or something. That's why.
So I'm sorry, I'm not familiar with that case. I am, the evolutionary biologists cite this case more, this one of lactose intolerance. So you had a really good question, and
maybe I can find the information about it and send it to you.
Yes, that would be wonderful. That would be wonderful. So just in the interest of time, can we, by the way, I really wanted to thank you for your patience when I was taking care of.
Oh, yeah, yes, of course.
We started late. So we've been going about an hour, 10 minutes, and I've saved really the worst part for the last, because it is really technical. I'll try to do it justice by at least referring to the literature. So you asked question number 3, how do you figure out if some mutation is slightly deleterious? How do we figure out if some mutation is slightly deleterious? So
yes, because,
independent.
may I ask?
Yes.
I would like to add to this one because I just wrote it very fast. So what I wanted to say, so in the model where you consider there is slightly deleterious mutations would accumulate and are not filtered out, so it would be better that you can evaluate what exactly these mutations are. Means to have some sort of information or function about how to evaluate genes and assign to the human, for example, genes its fitness, and then you know like this and this and this gene have this score, because it would be better, because if you cannot link the real genes to the model, this model is sort of floating in there. It cannot point to the real events what are happening in the world. This is my advice to this model.
Okay, so it's all right. First thing I wanted to, I should like to point out, let me try to get rid of some of these extra windows here so I don't get so confused. So the first thing near neutral theory. So the first thing I'd like to point out is the idea of nearly neutral or slightly deleterious. That's not a creationist term. So that's the first thing. That's not a creationist idea. It came from population geneticists, Tomoko Ota, and she was a student of Moto Kimura. So these are very, very respected evolutionary biologists. The formal term would be population geneticist. So when we say slightly deleterious mutation, I just want to point out this is not a creationist term. This is a term that evolutionary biologists have a kind of their own definitions for. And that's what makes it a little difficult, because then we have to go into population genetic literature. But let me show an example of a peer-reviewed paper that uses the concept. Kondrashov. And so I point the readers to this paper by Alexei Kondrashov, who was a colleague of my boss at Cornell University. And it says, this is the article in the journal Theoretical Biology, 1995, August 21st. So I'm just, for those who are interested, I'm just trying to give the references. And this is a very technical abstract, but I just want to, just for the sake of completeness, I'm just going to read a little bit from the abstract. It is well known that when S, the selection coefficient against the deleterious mutation, is below approximately 1/4 NE, where NE is the effective population size, the expected frequency of this mutation is approximately 0.5 if forward and backward mutation rates are similar. Thus, if the genome size G in nucleotides substantially exceeds the NE of the whole species, there is a dangerous range of selection coefficients, 1 divided by G less than S less than 1/4 NE. Mutations with S within this range are neutral enough to accumulate almost freely, but are still deleterious enough to make an impact on the whole genome. So this is the idea of the slightly here, it actually uses the word here. And he's saying several possible resolutions are considered, including soft selection and synergistic epistasis among very slightly deleterious mutations. So where I was headed with this is, you could see when you ask this question, if some mutation is slightly deleterious, you could see just by me sharing that abstract that this is like a can of worms to get through. I will try to explain it. If you want to have another talk on this in more detail, we can, but I'll try to cover maybe in 10 minutes kind of the idea. So this is not what I want to emphasize, this is not a creationist idea. This is something evolutionary biologists have defined themselves. And so this is really kind of, let me see if I have it here. This is a little bit, let me see how we can define a slightly deleterious mutation. They actually give, let me see if they have it, 1/4 NE. I want to see that 1/4. All right. Actually, Kondrashov had a better description. A slightly deleterious mutation, we might define it in terms of this so-called S coefficient that's less than 1/4 the population size. Now, let me just try to back up a little bit. Just hypothetically, just so we can kind of understand the concept. If we had a male and female parent, so this is a hypothetical species. This would not work with real species, but this is hypothetical. Say we had a male and female parent, and then they had 2 children, one male and female. Again, this is purely hypothetical. Selection could not possibly operate because there's only a choice of those 2 kids. I mean, selection will not be in play. Now, if they had a little bit more children, we could start to have a little more selection. So let's say we have this hypothetical population and they had, say, 10 children or 20 children, just give it some number. But if they have thousands of genes, one can only select for a few genes at a time. You can't select for all the genes simultaneously. So let's say, for example, we have one child that has a trait that enables him to run really fast, and then another child that has a trait that enables him to be smarter. So let's say a lion comes along and it's gonna, the fast kid will be able to get away, the smart kid won't, even though he has a good trait of intelligence.
The smart kid would not get in the situation where he's near the line.
Well, good point. Then there's other things like, say, just random accidents. Sometimes someone with a favorable trait, we know people that die of just an accident, had nothing to do with whether they had
Yes,
good
but
quality.
in population genetic, we view the genes in entire population, not the small numbers. So the same mutation appears in the population for the different parents. Like in time, this is the frequency of this mutation, and then they are selected either against or beneficial.
So
it's like that. Sorry.
That's how we generally think of it.
Yeah.
But there's some subtleties to this. You probably have heard the term mutational drift. And
Yes.
And what this is trying to say is that there's a point when something may be selectively advantageous. S is the measure of that strength of selection. So if it's just slightly advantageous, so what do we mean by slightly advantageous or slightly disadvantageous? Someone could have slightly weaker eyesight than someone else. They're obviously live. And we have obviously many people that have not the best eyesight over the course of their life, and some people that have better eyesight throughout all their life. And yet we see these people still, we see people with kind of maybe what we would call not optimal systems, still alive and reproducing. So kind of that in a medical sense. And so we have this definition, again, this is not a creationist thing. This was a definition that came up in terms of the population geneticists themselves. So it's helpful then to go into what an S coefficient is. And that's described on page, let me see what page this is, page 64. It could be actually earlier in Joe Felsenstein's book. And this is not a fun read. All I could tell you to understand.
I wanted just that you explain this formula. I view it.
I did before, but I didn't use S coefficients. So I was using the relative fitness with omega or W. I didn't use it with S coefficients. So it's page 57. It explains S coefficients. So in our first talk, I was just talking about this mean fitness here. But now so much of the literature uses this S coefficient. And this is, I'm just going to refer to the reader in this book that I reference a lot by Joe Felsenstein, Theoretical Evolutionary Genetics. It explains the S coefficients in brutal detail. But just suffice to say, the smaller the S coefficient, the smaller the selective advantage or disadvantage, just in qualitative terms. And then what Kondrashov does, because this is how you asked the question, how do we measure or how do we know what something is slightly deleterious or not? What is kind of like unfortunate, and this is why I had some problems with the definition, the evolutionary definition of fitness, is what will make it, I'm sorry, I'm having to page through all this. So page 57 will go into S coefficients. There, these S coefficients. What makes it, to me, just personally frustrating is it's slightly deleterious is dependent on the number of people in the population. So the number of individuals in the population determines whether something is slightly deleterious or not. So if you have a very small population, then there are more slightly deleterious mutations. If you took that same population and all the alleles or whatever, and you had a very large population, then they're less slightly, the threshold for slightly deleterious changes. It could be the exact same gene that's in question, or the alleles to that gene, to be more specific. But just because the population size has changed, the definition of slightly deleterious is different. What qualifies, I'm sorry, that's kind of like, yeah, go on.
We have now pretty big population size, so relative to many kinds of animals, I would say. billions people.
Oh, Peter, can you speak into the microphone more? I'm really sorry.
Okay, so I would say that the human population have now pretty big population size. Don't you think so?
Well, see, there's also a problem. It's what they call stirring, whether they can interbreed and stuff. So we might actually have very small, isolated. See, this is where some of the math in the definitions, and you probably, since you watched I thank you for watching my debate with Dapper Dino. This is the problem I have with these definitions, is that they, there's so many qualifiers, and it makes it hard to determine what a slightly deleterious mutation is according to evolutionary definitions. I don't even try personally to use their definitions. And I mean, I have to at least understand it enough since I read the literature, but
I meant it in the context of genetic entropy model.
Oh yeah,
because
so, sure.
I think there is some models like, for example, one mutation cannot be selected again, so beneficial because it's very small. But if organisms have organisms have many genes, and many genes add a little bit, become more or less, and this is how entire organism have a significant score for selection, either against or beneficial.
And let me see if I.
So because we have lots of genes and lots of variation of them.
So, okay. So no, I'm glad you asked. So in the context of creationist theory, specifically the genetic entropy theory, I'm going to bring up a slide that we had in our last talk. I hope I still have it. I'd be, if you give me a few moments, I'm going to try to bring it up.
Okay.
And see. Motors limit. Let's see if I can bring it up. So in the context of the genetic entropy theory, yeah, let me bring this. Okay, so this was a slide from our last talk, and I was just pointing out that if every child on average has a defect, there is going to be what we might call a slightly deleterious mutation, in the sense that maybe one defect is really severe for one child, which is really kind of sad. I don't want to make light of this. I have good friends that have horrible genetic diseases that are just terribly debilitating, and one of my dear friends is terminally ill. So I don't want to make light of this, but relatively speaking, some mutations are not quite as bad as others. So if natural selection came along and started eliminating, can't possibly eliminate everyone. So we'll just take maybe a fraction of them and not eliminate them. So that's how I would define slightly deleterious. So relatively speaking, let's
okay. May I ask? So
sure.
So don't you think that if mutation is slightly deleterious, it is because of the, so to say, biochemistry? How does gene interact? And this gene can, in other circumstances, become slightly beneficial because further of some, maybe there is some other mutation in the organism that causes that. And so my question was about that. So can you for sure say that some mutations are slightly deleterious without any further factors? Or it is sort of dependent of other factors and interaction with other genes, and that is
yes. Well, I think you raised a good question. I think it's very hard to define things. We can say that some mutations are almost definitely more severe than others in terms of, I mean, we just see it from a medical standpoint. Some are very much more damaging than others. There are others that it seems we can tolerate. We have some experimental evidence of this too.
Maybe do you have example of slightly deleterious mutations?
Yeah.
Yes,
This experiment, I'll show you some experimental evidence of this. So her name is Brenda. Okay, it's easier to do it with yeast because we really don't want to do too many experiments on live humans. So I hope you'll forgive me for citing yeast, but medical researchers, the last organism they want to do an experiment on is a live human. There are lots of ethical concerns. So
yes, but you do not do experiment with human. You just need to evaluate what this gene does in human body, and that is sort of okay.
So this Brenda Andrews did research on this to show slightly deleterious mutations. She doesn't use the word, but she actually had experiments relevant to this, and she was invited to a special planning conference at the NIH regarding human health because her experiments were pretty important, even though it was on yeast. And let me see, she had a really cool project. Wow, she's doing so much now. Brilliant scientist. I'm sorry, it's been a long time since I visited her website, but she's, oh my goodness, she's long since moved on to other, she's done so many, she's just a brilliant researcher. But maybe I could highlight, so this would have been way back. Oh my goodness. I apologize. I'm just going to refer the readers to the website, and then just maybe explain what she was trying to say. There is this little picture she has here. If I could expand it. I know that may not be very meaningful, but what she did was she took yeast, and this is a constellation of various yeast genes, and she was knocking them out, disabling them. So she found out that she could knock out some genes. If she knocked out just one, the organism would still reproduce. It didn't have an effect. And she was actually surprised. She was able to knock out so many genes and the organism survived. So I classify that as slightly deleterious. However, when she started knocking out genes in pairs, so she'd knock out one gene in one cell line or yeast line or whatever it was, and then take that line and then knock out a second gene. And she would knock out all the other, she'd methodically go through each of the genes in the list of all the genes in the yeast, because yeast is a simple organism. We can actually do this. Don't even try it for a human. And she found out that second knockout really just compromised the organism severely. So we have an experimental example of where the organism it's just like a tire wearing out. It can take a little bit of tread loss, but there comes a point it starts to accumulate, and it's more than you have to get the tire replaced. It's kind of similar in the case of her experiments. The organism could take some gene loss, and at the time it only had slight effect on its ability to reproduce. But as we added more and more of these mutations, it really started to affect it. So in that sense, it is slightly deleterious. And when we're dealing with very small populations, like say eukaryotic animals, when you have very small populations that are they're small, I don't know if it has to be well stirred or not, and that's apologies to the viewers. I don't know the immediate question to that. Well stirring means that the population is able to freely mate, whereas that's not really the case because people are geographically isolated. That's a whole another can of worms. But in any case, if the population is small, then it's very possible that an individual with a slight deleterious mutation could end up dominating the population. His genome is passed on. A lot of his traits are passed on just by accident. We call that genetic drift. And that's what Kondrashov's paper was concerned about, is that these slightly deleterious mutations could just freely accumulate over time. And that's what I was, I didn't have to use the definition of slightly deleterious to make this argument. It's like, well, it didn't matter if it's slightly deleterious or not. I didn't have to define it. All I had to do with Muller's limit is to show that if most of the gene mutations are damaging and that they're beyond the limit of one mutation per individual per generation, that let me see if I had it. As Kondrashov said, did I have that picture? Let me see if I can get it. I had a picture here. Apologies if I didn't have it. No, I don't have it in here. That it would accumulate freely over time. It didn't matter how one was defining slightly deleterious or not. The only thing one had to be concerned with was this equation here, and that if U is over 1, which it is, U is estimated to be between some very generous estimates being only as low as, like, say 10, but all the way to 100. And that was, so in answer to the question, how do we figure out if a mutation is slightly deleterious? And my answer is, it's too hard. I don't try to figure it out, and evolutionary biologists have a hard time pinning it down. So it's really, I can't answer it. And what I tried to say here is, we don't need to define slightly deleterious. It's just the term. What we really need is this figure U, which is the number of mutations per individual per generation. So it doesn't matter if it's slightly or more. The important thing is if U is over 1, when we get up to 10 or even 100, it starts to become very difficult to justify why that we're actually improving. And I think that that's one of the reasons every major geneticist that I know of doesn't think that our genome is improving, and they're particularly concerned about the decline of intelligence and mental health. So I'm sorry if you wanted me to define it. I probably didn't do a very good job because I don't deal with it, and I don't think it's needed. But there is literature that describes it. So we've talked for about an hour and a half, and my voice is getting raspy. I think you had really great questions. I can't tell you how much I appreciate this interaction, because it's really not fun doing a total monologue. It's kind of nice to be asked questions, and it gets me to think, and it helps me know if I'm communicating things poorly or well. And it helps me improve the way that I understand things. So if you wanted to talk about another topic sometime, if I can fit you in my schedule, I will. Everyone now is starting to try to get a hold of me to talk, which is nice. I did not ask to debate Matt Dillahunty, but that just came up. I was sought after. So, I mean, it's kind of nice to know people want to hear me talk. But doing this sort of discussion like what we did today, I enjoy this more than kind of these debates on religion.
Yes, I enjoyed it too. Thanks.
Yeah.
So I have still other questions to you, but we'll do it later.
Yes, maybe you can, if you can type them up and email them to me, because that will also give me a chance to think about it until we meet again, because then I can give you a better answer and I can start looking up things. And at the very least, if I don't have a good answer, at least I can say, okay, I looked it up, Peter, I don't have a good answer. So I will try to tell you and the viewers when I don't think I have a good answer to a question. And one example is that sure.
Are you familiar with Python programming language?
Yes, a little bit. I used to be a C programmer but now I'm trying to learn.
So if say I give you some program in the Python about something, so are you able to see that what it is doing and use it somehow? Yeah.
I'm not good enough to do that, but if you would like to help teach us what your program does, I would be very, very personally grateful.
Mm-hmm.
So if you show it and then I ask you and you tell me what this does, I think I'd be able to understand. I would learn. That'd be a great way for me to learn.
Yes.
So if it's not longer than say 2 pages, a 3-page Python program would probably be more than I could digest. But I'd be personally grateful. So if you want to review that, we can try. If we fail,
okay.
If the show doesn't work, at least we tried.
Okay, I make a sort of tutorial so people understand what's happening. So, and I wanted to ask you, what do they calculate in this program? What was its program's name?
Oh, Mendel's accountant?
Yeah, Mendel's accountant. Yeah. So can you explain short how does they calculate this stuff? So
yeah, it was used to in the process of creating certain peer-reviewed papers that got published. And because it was critical of evolutionary theory, it's been challenged by people in other people in the mainstream. So they were complaining that the Mendel's accountant wasn't accurately modeling biological systems. And the software, there's really a very sad story about the software. The latest version was written in a language called Go. He was a very good software developer, but he died of cancer. And so the program has not been maintained. There's been about 3 or 4 versions of it. The first being written in Fortran, then the next, I think, in Java, and then the next in Go. And it's been discontinued. The reason it's been discontinued is the questions that it addressed, at least now in the peer-reviewed, a lot of peer-reviewed literature independent of Mendel's accountant are affirming the claims that were made using Mendel's accountant. So what I mean by that is Michael Lynch is also saying kind of the same thing about this mutational meltdown model, etc. So it would be, I've said, rather than criticize Mendel's accountant or try to rebuild it, just take Joe Felsenstein's book, this one here, and just write a program that will implement his evolutionary theoretical genetics. If we do that,
so this from this book entirely.
Yeah, this is from an evolutionary book, and he actually gives computer examples that can show it. This is why I like this book. I mean, trust me, this is a painful read. This is not a very nice book. I mean, it's a nice book, but it's just, when I say it's not a very nice book, I'm not trying to say it's badly written. It's just, the material is just very laborious. And he even has, we could, for example, do the Fisher-like theorem here and model that with a computer. I said, this is within the range. And then there are examples that Mendel's accountant could actually handle the deal with linkage, and where this was really important. There's one, let me see if I could find it. Let's see, Moran. There was one example that Mendel's accountant could handle that even Joe Felsenstein's book where it talked about where fitness doesn't ever maximize. And it would take something the power of Mendel's accountant to do this. So I say, let's just build a model independently that everyone will agree with. So we could take someone like Dr. Dan or any other mathematician and then a programmer and say, okay, we have all the predicted results here in Joe Felsenstein's book. So if the program can't replicate the results, and he had lots of numerical examples, and you're supposed to get certain amounts, certain kinds of results. If you don't get these results, then at least according to population genetic theory, there's something wrong with your program. So that's what I would recommend. And and what has happened, interestingly, Felsenstein was saying that most of the genome has to be junk because of his calculations. He even said so in his book. We can actually simulate that numerically, not just analytically with all the equations. We could just throw all this in there and say if it has these
may
parameters.
I ask, is
Sure.
it possible to make a real model about human genome? Like model all kind of gene and what the
No, that's a good question. The problem,
fitness of those
the problem I had with population genetics is it doesn't know how to model the complexity of the gene. That was my
Yes.
debate with Dapper Dino. I said, if all you're doing is just saying this one will make it more reproductively successful, a little bit here and there, one little environmental change and all those coefficients that you're using, like those S coefficients, it's out the window. And that's a known problem. And that's why that's why I had that debate. I said some population geneticists have basically bailed out of the whole industry because this whole thing with the S coefficients, one is assuming that it is relatively, or most of the equation models are the S coefficient is constant over many generations, and everyone knows that can't be the case. But because it's the only way we can model it and still have any hope of solving things mathematically, that's what they use. But we know that that assumption is kind of faulty. But if one wants to still throw these numbers in there, because the S coefficients determine if it's slightly deleterious or not. That's how we just define it, as I said. But we know that these S coefficients can either go up or down depending if the environment goes changes. Like in the example of the cavefish, a beneficial trait, like say eyesight, it goes into a cave, it's now a deleterious mutation. And that's why I had a real problem with defining fitness in terms of reproductive success. But if we want to use the evolutionary definitions, we can take a book like Joe Felsenstein and then model those parameters. But you asked, can we model the human genome? I'll say absolutely not. Not in any way that's meaningful. So this is kind of the problem that even the creationist community has had to deal with, is that they're still using evolutionary definitions, which are still, even by evolutionists' own admission, it's problematic. Lewontin being like the forefront. And then there have been papers in 2018. Just for completeness, I'm going to share, this was a real eye-opener for me. I'm going to just share this paper, Grod Wall. This was talking about population genetic theory, and I wish I had had time to discuss it in my debate with Dapper Dino. This is published July 6, 2016. The theory was beautiful indeed. Rise, fall, and circulation of maximizing methods in population genetics. And this is talking basically about how the whole field went through a real crisis. Maybe I mentioned Lewontin. He said, it said Lewontin became the staunchest critic of maximizing principles in evolutionary biology. And this is just a compelling read. So I just point our readers to that. So back to your question about, can we model the human genome? No one can model it. If we could, honestly, if we could model the human genome, we probably would be able to cure all diseases by now. I mean,
okay, so what information do we need to do that? Just can you tell it so?
To model the human genome?
Yes. What information do we need to model it? Like we have DNA strand.
Yeah, I think
its functionalities and.
Yeah, that's a very good question. The problem is we don't even have the data. We hardly even know what each of the genes do, and even genes that are well. I'm sorry.
We have sequenced human genome, and we know functionality of a lot of genes.
We don't know it in detail. And the reason I'm saying that is I happen to work with someone who studies the topoisomerase 2 family of enzymes. And even then, we have people working this for practically their whole professional career. We still barely understand how it works. And that may seem surprising. Let me show. I mean, one may think it's trivial, and I can understand why people would think it's trivial. Let me just show the, for the benefit of the reader, just the spelling of the topoisomerase 2 enzyme. It's deceptively, it may look like a simple problem, but it's way harder than we think. And let me show why. Let's see if I could find it here. Yes. Okay, so this is the, I mean, it's really, it's almost surprising. This is the spelling of the topoisomerase 2 enzyme. It's about 1,500 letters. So any human being could type something. I mean, this is just like about the size of a paragraph. Why does it take researchers an entire professional career to understand just all these letters and what they do? It's because when it folds into a three-dimensional conformation, and it's starting to interact with the cell, many of these molecules here will have dynamic changes, meaning they get chemically modified, and they're flipping on and off like bits. We call them post-translational modifications. We don't know how this actually works. If we did, we'd probably be able to cure cancer. So, yes, we
model it biochemically.
We cannot model it chemically. We can model only parts of it. It is that difficult. And so if the pharmaceutical companies would like to be able to understand how to control this, and we could make better chemotherapies. In fact, that was the, let me see if I have it. If you'll give me a moment, I'll show you the publication that I actually, it's actually a peer-reviewed abstract. It's not a full-up publication, but I mean, this one means a lot to me because I had worked in the defense industry where a lot of my work was not, we would not be allowed to share it with the public. So it's nice to be in the biology community where we're able to share things more freely and encouraged to. So this is the name of the journal, and you could see my name right here. I was just one of several co-authors, one of like 10 co-authors. And I'm going to show something that shows a little bit of the complexity of, so that's the topoisomerase molecule. And this is the human topoisomerase here. And we're trying to figure out the complexity here because they're all, you're asking about the chemical interactions. Everything where you see an arrow is where there's a chemical modification. And these are like little bits being flipped on and off in real time. What
modification are these?
Yeah, these are
charge or something.
I'm sorry?
This electrical charge, this modification, or what it does?
It's what they call a chemical modification. It's like, in this case, if it's purple, it's what they call a phosphorylation. So it adds not an electrical charge per se, but a phosphorus molecule. And then in here, it would add another kind of molecule that they call a simulation molecule. And let me show you, let me see if I have another diagram here, because I'm very grateful for your curiosity into these topics. I think I have maybe one diagram. Let me see. Topo. Yes, I'm gonna show. So we have all these chemical modifications. Let me just make this bigger. So this was outside of the paper because we couldn't fit it, or the poster session. But these shows all these, everything that you see here is where it can modify. So this is like a computer bit. the protein can actually act like a computer memory, and we can see all the locations where it is being modified here. And we don't know what this code means. That's why this has become a much more difficult problem. So when you're asking, can we model the genome? I'm just like, there are too many things we don't know to be able to model it accurately. And because there are about 150 of these bits in the topoisomerase molecule itself. And if we export all the combinations, that would be what, like 2 raised to the 140th power just to understand it. So this is, maybe that's a long way to explain why I don't think we have the capacity to model it with any fidelity. If we could, we probably would have cured all human diseases by now. So there's not going to be, the National Institutes of Health spends $40 billion a year on researching biology, and then we add the biotech. There's going to be a lot of research. We're not going to be able to model it. So, yeah, it kind of looks. I mean, that's why population genetics was appealing. Is they like to simplify biology, and it's nice that they could build out theories on simplistic ideas because the math is easier. We could run it on computers, but the problem is it doesn't capture the real complexity of biology, and for that reason, that's one reason I tend to spend more time not with things like Mendel's accountant or population genetics. I prefer spending time with the protein science, and even then only like maybe 5 or 6 proteins, because I think I like the idea of being a little bit more thorough and accurate in understanding how biology works. So the population genetic models are just too simplistic for my taste. So even though you hear a lot of creationists cite genetic entropy, I don't use it quite in the same way that they do. The way that I use it is like when in our talk on Muller's limit. So I'm sorry my voice is running out. I would love to continue this conversation again. So if you're able to come up with some questions, maybe in a couple of weeks we'll have this. I just want to tell you that our the sessions and shows that we've had, those have been some of my favorite because they're very technical.
Yes. Okay, so thanks a lot. It was really great, and we see you later.
Yes. Take care, Peter.
Yes, you take care too. Merry Christmas and Happy
Yes,
New Year.
Merry Christmas and Happy New Year.